Hello, and welcome to another gentle journey on Sleepy Professor Stories. Tonight, we turn our attention inward towards something you carry with you every moment of your life, yet rarely pause to consider in stillness. The human brain rests softly inside your skull, about the size of two open hands cupped together. It weighs just over a kilogram, light enough to hold, yet complex enough to contain your memories, your thoughts, your sense of self. It does not glow or hum loudly. It does not demand applause. It simply works patiently and continuously, like a night gardener tending unseen pathways. Even now, as you settle into a comfortable position, your brain is quietly active. Billions of cells exchange tiny electrical signals. These signals are not flashes of lightning, but gentle pulses, brief changes in voltage that ripple along delicate fibers. Each pulse lasts only a fraction of a second. And yet, through these small changes, you see, you hear, you remember, you feel. The brain is composed of soft tissue, protected by layers designed with remarkable care. Bone surrounds it like a curved shell. Beneath that shell lie three thin membranes, called the meninges, which cushion and support it. Between these layers flows clear fluid, cerebrospinal fluid, which acts like a calm inner tide, absorbing gentle impacts and carrying nutrients. You do not have to manage this. You do not have to supervise it. It happens quietly, whether you are awake, dreaming, or fast asleep. As we move through tonight's journey, there is nothing you need to memorize, nothing to achieve. Think of this as a slow walk through a landscape you already inhabit. We will wander through memory, emotion, dreams, learning, creativity, and the peaceful rhythms that guide thought. Not in a technical or hurried way, but in a way that feels spacious and calm. Your brain is often compared to a universe, and in some ways the metaphor fits. It contains roughly 86 billion neurons, each one capable of forming thousands of connections. If you could shrink down small enough to travel between them, you might imagine a vast forest of branching trees, their limbs reaching and intertwining. Signals pass from one to another across tiny gaps called synapses, where chemical messengers drift like soft whispers in the dark. These chemical messengers, neurotransmitters, move in measured quantities. A small release can influence mood, focus, or calm. Dopamine helps guide motivation and reward. Serotonin contributes to steady mood. GABA slows activity, creating a sense of relaxation. Even now, as you listen, these molecules are flowing, adjusting, balancing. There is no need to direct them. Your body has practiced this coordination since before you were born. In fact, the brain begins forming early in development, starting as a simple neural tube just weeks after conception. From that narrow structure, the cells multiply and specialize. Regions take shape, pathways form. By the time you are born, your brain already contains most of the neurons you will ever have. What changes over time are the connections between them, growing and reshaping in response to experience. This ability to change, called plasticity, is one of the brain's quiet strengths. It adapts gently, like a river adjusting its course over years. The surface of the brain is folded into ridges and grooves. These folds are not random. They allow a large surface area to fit within the limited space of the skull. If the brain was smoothed out flat, it would cover a much wider area. Instead, it curves inward and outward, forming patterns that are both efficient and uniquely yours. No two brains fold in exactly the same way. Just as fingerprints differ, so too do the subtle contours of your inner landscape. The brain is divided into two hemispheres, left and right, connected by a thick band of fibers called the corpus callosum. These fibers allow information to travel between sides in milliseconds. While popular culture often speaks of a logical left brain and a creative right brain, the truth is softer and more integrated. Most activities involve cooperation across both sides. When you speak, solve a puzzle, imagine a memory, or feel emotion, networks span the entire structure. The brain prefers collaboration. At the back of your head lies the occipital lobe, devoted largely to vision. Light enters your eyes. It is converted into electrical signals and travels along the optic nerves to this region. There, patterns of brightness and color are interpreted. What you experience as a seamless image is actually a carefully constructed perception assembled in fractions of a second. Along the sides are the temporal lobes, involved in hearing and aspects of memory. When someone speaks, vibrations in the air reach your ears, and signals travel inward, becoming meaningful sound. Words take shape. Meaning forms. Now, in the upper regions, the parietal lobes help you sense touch and spatial awareness. They allow you to know where your body is in space, even with your eyes closed. And at the front rests the prefrontal cortex, an area associated with planning, decision-making, and self-control. It matures gradually, continuing to develop into early adulthood. This slow development is part of why patience and long-term thinking strengthen over time. Growth here is not rushed. It unfolds steadily, shaped by experience and reflection. Deep within the brain lies the brainstem, an ancient structure that regulates breathing, heart rate, and basic survival functions. You do not have to remind your lungs to expand. You do not need to instruct your heart to beat. The brainstem manages these rhythms automatically. Even as you drift towards sleep, it remains attentive, steady as a metronome. If you like listening to calm explorations like this, you are always welcome to support the channel in whatever small way feels natural. Perhaps by subscribing or sharing with someone who might enjoy a gentle evening journey. But there is no expectation here, no urgency, only quiet gratitude for your presence. For now, let your shoulders soften. Let your jaw unclench. Notice your breathing, slow and unforced. There is nothing you need to prove to this moment. Your brain has carried you through countless days already. It will continue its patient work through the night. As we move forward, we will look more closely at how its cells communicate, how tiny electrical signals become thought, memory, and feeling. Because within this quiet universe, a vast forest of neurons is waiting, gently illuminated by flickers of living light. And that is where our journey continues next. If you could gently step inside your own head, not in a dramatic way, but in the soft spirit of curiosity, you would find no sharp edges, no rigid machinery. Instead, you would discover something surprisingly delicate. The brain is made of soft tissue, with a texture often compared to firm custard or tender tofu, protected carefully within the curve of your skull. It rests there without complaint, floating in a clear cushion of fluid that absorbs movement and distributes nutrients, as steady and quiet as a sheltered pond. The skull itself forms a strong outer boundary, a protective dome shaped by millions of years of gradual change. Beneath this bone lie thin protective membranes, layered gently one over another. These membranes cradle the brain, holding it in place while still allowing subtle movement. Between them flows cerebrospinal fluid, circulating slowly, bathing the tissue in nourishment and carrying away waste. You never feel this motion. It happens continuously, like a tide that rises and falls without announcement. The brain's surface is not smooth. It folds in on itself in graceful curves, forming ridges called gyri and grooves known as sulci. These folds allow more surface area to fit within the limited space of your skull. If the brain were unfolded completely, it would stretch far wider, exposing a vast sheet of processing power. Instead, it gathers inward, compact and efficient, like a landscape of hills pressed gently together. Each fold increases capacity without adding weight, a quiet example of nature's economy. You carry about 1.5 kilograms of this living structure. It makes up only a small fraction of your body weight, yet it consumes a remarkable share of your energy. Even at rest, when you are lying still and breathing softly, the brain uses about 1/5 of your body's oxygen and calories. It does this not because it is straining, but because its cells are constantly maintaining balance, preserving electrical gradients, and communicating across microscopic distances. The brain divides into 2 hemispheres, left and right, mirrored but not identical. These hemispheres are connected by a thick band of nerve fibers called the corpus callosum. Through this bridge, signals pass in milliseconds, allowing both sides to share information. You do not experience your mind as divided because this connection is seamless. When you reach for an object, interpret a sound, or recall a memory, both hemispheres collaborate quietly. Within each hemisphere are regions known as lobes, each associated with certain functions. At the front rests the frontal lobe, involved in planning, decision-making, and aspects of personality. It helps you pause before acting, consider consequences, and shape intentions. Behind it lies the parietal lobe, where sensations from your body are mapped and interpreted. When you feel the softness of fabric or the warmth of a blanket, signals travel inward and are gently translated here into awareness. Toward the back sits the occipital lobe, devoted primarily to vision. Light enters your eyes, becomes electrical signals, and travels along the optic nerves until it reaches this region. What you experience as a single image is the result of countless tiny calculations happening almost instantly. Edges are detected, colors sorted, motion tracked. Yet it feels effortless to you, as if seeing requires no work at all. On the sides, near your temples, are the temporal lobes. These areas help process sound and contribute to memory. When you hear a voice, vibrations move through the air and into your ears. Mechanical motion transforms into electrical patterns that travel inward, where meaning slowly emerges. Words are recognized, tone is interpreted, memories connect. All of this unfolds in fractions of a second, but it does not feel rushed. It feels natural. Deeper still are older structures. It's shaped long before human language existed. The brainstem at the base regulates breathing, heart rate, and basic rhythms of survival. It operates continuously, even while you sleep. You do not need to remind your lungs to expand or your heart to beat. These cycles continue in gentle repetition, like waves brushing the shore. Nearby lies the cerebellum, tucked beneath the back of the brain. Though smaller in size, it contains a vast number of neurons and plays a key role in coordination and balance. When you walk across a room or lift a cup, it refines your movements, smoothing them so they feel fluid. Without it, motion would become unsteady. With it, actions blend into graceful continuity. The brain's outer layer, the cortex, forms the wrinkled surface you might recognize from diagrams. This thin sheet of gray matter contains densely packed neurons responsible for perception, thought, and voluntary movement. Beneath it lies white matter, composed of long fibers that connect distant regions. The contrast between gray and white is not about color alone, but about function, processing and communication woven together. Blood vessels thread through every part of this structure, delivering oxygen and glucose. The brain depends on a constant supply of both. Even a brief interruption can cause cells to struggle. That is why circulation is so carefully regulated. Arteries branch into smaller vessels, spreading outward like the roots of a tree, ensuring that each region receives what it needs. There is no single command center where everything happens. Instead, the brain operates as a distributed network. Different regions specialize, but they rely on constant communication. When you solve a problem, emotional centers, memory systems, and decision-making areas all contribute. Activity flows across pathways in shifting patterns, like wind moving through tall grass. As you rest now, perhaps noticing your breathing, your brain remains active in ways you cannot feel directly. It adjusts chemical levels, refines connections, and monitors your internal state. There is nothing you need to guide, nothing to correct. This architecture has been shaped over millions of years of gradual refinement, adapting to changing environments and growing more complex with time. Each part fits within the others, not as isolated compartments, but as interwoven systems. Structure and function blend seamlessly. Form supports activity. Activity reshapes form. It is a living architecture, responsive and adaptable, yet stable enough to carry you through each day. If you allow your thoughts to soften, you might imagine this entire structure resting in quiet darkness, gently illuminated by tiny pulses of activity. Not frantic, not chaotic, but steady and purposeful. The folds and curves, the layers of protection, the hemispheres in conversation, all working together without demanding your attention. And as we linger here, appreciating the calm design of this inner landscape, you may begin to wonder how these countless cells actually speak to one another, how silence turns into signal, and how a soft electrical whisper becomes the language of thought. If you could shrink yourself down to the smallest imaginable scale and wander gently through the living landscape inside your head, you would find yourself standing in something like a forest at dusk. Not a forest of wood and leaves, but of delicate branching cells, stretching and intertwining in quiet complexity. These are neurons, the primary messengers of the brain, and there are tens of billions of them, each one shaped with fine extensions that reach outward like the limbs of ancient trees. Every neuron has a cell body where its essential work begins. From this body extend thin branches called dendrites, spreading outward to receive signals. A single longer fiber, known as an axon, carries signals away. Some axons are short, connecting nearby neighbors. Others extend remarkable distances, linking regions far apart. If you were to follow one with your tiny steps, you might travel from the front of the brain to the back in a single slender line. Neurons communicate using electrical impulses. These impulses are not dramatic sparks, but brief shifts in voltage that travel along the length of the axon. Inside each neuron, charged particles move in carefully controlled patterns. When the conditions are just right, a rapid change occurs, and a wave of electrical activity sweeps forward. This event lasts only a fraction of a second, yet it carries meaning, like a whisper passed along a chain of listeners. The speed of this electrical signal depends on the structure of the axon. Many axons are wrapped in a fatty insulating layer called myelin. This sheath allows impulses to travel more quickly and efficiently, hopping between tiny exposed gaps. In well-myelinated pathways, signals can move at impressive speeds, allowing coordination and perception to feel almost instantaneous. You lift a hand, and it responds without delay. You hear a sound, and recognition follows almost immediately. Yet neurons do not physically touch one another. Between the tip of one axon and the dendrite of the next cell lies a microscopic space called a synapse. Here, communication shifts from electrical to chemical. When an electrical impulse reaches the end of an axon, it triggers the release of tiny molecules stored in small sacs. These molecules drift across the narrow gap and bind to receptors on the receiving neuron. These chemical messengers, known as neurotransmitters, fit into their receptors like keys into carefully shaped locks. And some encourage the next neuron to fire its own electrical signal. Others make firing less likely, creating balance within the network. Excitation and inhibition flow together in delicate proportion. Without this balance, activity would either fade into silence or surge uncontrollably. Instead, the brain maintains a steady rhythm, adjusting continuously. You might imagine these synapses as small clearings in the forest, where signals pause briefly before continuing on. In a single moment, one neuron can receive thousands of such inputs from different neighbors. It integrates them quietly, summing and weighing each influence. Only when the combined input reaches a certain threshold does it send its own signal forward. This process unfolds countless times each second, across billions of cells, yet you are not aware of the individual steps. You simply experience thought. Neurons are not all identical. Some are pyramidal in shape, with long apical dendrites reaching upward. Others are interneurons, shorter and densely connected, modulating activity within local circuits. Some specialize in sensory information, carrying signals from the eyes, ears, or skin toward the brain's interior. Others are motor neurons, sending commands outward to muscles. Each type contributes to a vast and layered conversation. The connections between neurons are not fixed in stone. They change with experience. When two neurons activate together repeatedly, the connection between them can strengthen. This strengthening occurs through subtle changes at the synapse, increasing the likelihood that future signals will pass along the same route. In this way, patterns form, habits develop, memories take shape. Learning is not a separate structure added to the brain. It is a gradual reshaping of the forest itself. At any given moment, only a small fraction of neurons are firing actively. The brain is not lit up all at once. Instead, activity moves in shifting patterns like fireflies blinking in different corners of a field. A region involved in vision may glow with activity when your eyes are open. Areas related to language may brighten when you speak or listen. Networks associated with emotion may stir in response to memory. The light is dynamic, always moving. Even in rest, neurons remain active. They maintain electrical gradients across their membranes, carefully controlling the movement of sodium and potassium ions. This maintenance requires energy, which is supplied by glucose and oxygen delivered through the bloodstream. Though the brain is small relative to the body, it consumes a significant portion of available energy, precisely because these gradients must be sustained. It is a constant, quiet effort, like keeping a lamp lit through the night. There is nothing you need to manage within this forest. The signaling, the balancing, is the strengthening and softening of connections. All of it proceeds automatically. As you breathe in and out, networks coordinate to regulate that rhythm. As you relax your shoulders, motor circuits adjust. Even your awareness of these words arises from patterns of neuronal firing unfolding gently in real time. Over years, experiences shape this network. Childhood brings a surge of new connections as the brain eagerly forms links in response to sights, sounds, and touch. Later, some connections are pruned, making pathways more efficient. This refinement continues through adolescence and into adulthood. It is not a loss, but a focusing, like trimming branches so the tree grows stronger. Neurons also rely on support cells called glia. Once thought to serve only as scaffolding, these cells are now known to assist in maintaining the environment around neurons, regulating chemical levels, and even influencing signaling. They help clear excess neurotransmitters from synapses and provide metabolic support. The forest is not made of neurons alone. It is an ecosystem, cooperative and interdependent. When you feel a memory surface or a thought drift by, it corresponds to specific networks activating in coordinated fashion. Patterns of firing repeat in familiar ways, giving rise to recognition. Yet the experience itself feels seamless. You do not sense the electrical pulses or the chemical exchanges. You sense meaning, image, emotion. And so, as you lie here in stillness, billions of neurons continue their quiet conversation. Signals rise and fall, connections strengthen and soften. A living forest hums beneath the surface of awareness. There is no urgency in it, only continuity. Soon we will turn our attention to the rhythms that emerge from this collective activity. The broader patterns that sweep across regions in waves, guiding states of wakefulness and sleep. For within this forest of neurons, something larger begins to pulse, steady and slow, like a tide preparing to move in. If the forest of neurons is the landscape of your inner world, then brainwaves are its weather. Broad, slow-moving patterns that rise and fall across wide regions at once. You do not hear them, and you do not see them, but they are present nonetheless, measurable as soft oscillations in electrical activity. When millions of neurons fire in coordinated timing, their combined signals form rhythms that can be detected on the surface of the scalp. These rhythms are not random noise. They are patterns, steady and shifting, like tides responding to unseen forces. When you are alert and focused, solving a problem or reading carefully, faster waves tend to ripple through the brain. These patterns are often called beta waves. They move at higher frequencies, reflecting networks engaged in active thinking and attention. It is not that the brain is strained in these moments. Rather, it is finely tuned, coordinating signals quickly across regions that analyze information and guide action. As your focus softens and you begin to relax, perhaps closing your eyes or letting your thoughts wander, slower rhythms often emerge. Alpha waves become more noticeable, especially in the back regions of the brain associated with visual processing. These waves are commonly observed when you are awake but calm and not concentrating intensely, simply resting in awareness. They carry a steady, unhurried cadence, like gentle ripples across still water. If you drift further towards sleep, another rhythm begins to appear. Theta waves, slower than alpha, become more prominent during light sleep and moments of deep relaxation. They are often associated with the borderland between waking and dreaming, when images may flicker softly behind closed eyes and thoughts feel less linear. In this state, the brain is not shutting down. It is reorganizing, shifting from external attention toward internal processing. And as sleep deepens, the slowest waves of all begin to dominate. Delta waves roll in long, powerful sweeps across large areas of the brain. These are characteristic of deep, restorative sleep. During this stage, neurons tend to fire in synchronized bursts, followed by brief periods of quiet. This coordinated pattern allows the brain to conserve energy and support processes that maintain cellular health. It is during these slow waves that the body often repairs tissue and strengthens immune function, guided in part by signals from the brain. These different rhythms are not isolated states, but fluid transitions. Over the course of a typical night, your brain cycles through stages of sleep in roughly 90-minute patterns. Light sleep gives way to deeper stages, and then to periods of rapid eye movement when brain activity becomes more varied and dream imagery can become vivid. Even in dreams, brainwaves shift, reflecting the unique patterns of that state. The cycle repeats several times before morning. Brainwaves arise because neurons influence one another. When large networks synchronize their firing, their electrical activity adds together, producing measurable oscillations. This synchronization can enhance communication between regions, timing signals so they arrive when neighboring neurons are most receptive. In this way, rhythms help coordinate information flow, much like a shared tempo allows musicians to play together smoothly. You may notice that certain mental states feel distinct: alert, drowsy, deeply asleep. Beneath these experiences, the shifting balance of brainwave patterns plays a role. Yet there is no need to control or direct these rhythms. They emerge naturally in response to light, darkness, activity, and rest. As evening arrives and light fades, signals from your eyes travel to a small structure deep within the brain called the suprachiasmatic nucleus. This cluster of cells helps regulate circadian rhythms, aligning internal cycles with the external day-night pattern. As darkness continues, the pineal gland releases melatonin, a hormone that supports sleep readiness. The rising levels of melatonin gently encourage the transition from alert beta rhythms towards slower waves. This process is gradual, unlike the dimming of a room at sunset. You may not notice the precise moment when wakefulness begins to soften into sleepiness, but the shift is unfolding beneath the surface. During deep sleep, the brain also engages in a kind of housekeeping. Recent research suggests that cerebrospinal fluid circulates more freely through brain tissue during slow-wave sleep, helping to clear metabolic waste products that accumulate during the day. This cleansing is subtle and continuous, supporting long-term health. Again, there is nothing you need to supervise. The rhythm of slow waves supports this process quietly. Even when you are awake now, listening calmly, your brainwave patterns are shifting gently. Perhaps alpha waves are present, reflecting a relaxed but attentive state. If your thoughts begin to drift and your eyelids grow heavier, theta waves may begin to mingle in. These changes are natural. There is no right rhythm to achieve. Your brain adjusts to your needs moment by moment. Scientists measure these oscillations using electroencephalography, placing sensors on the scalp to detect voltage changes produced by synchronized neuronal activity. The patterns appear as flowing lines on a screen, rising and falling in steady arcs. To a trained eye, these lines tell a story of state and transition. To you, they remain invisible, simply part of the background hum of being alive. And the remarkable thing is that such complex experiences, attention, imagination, sleep, can correspond to these gentle waves of electrical coordination. Not because waves alone create thought, but because they reflect the collective timing of vast networks working together. Rhythm shapes possibility. Timing shapes communication. And so, as you continue to breathe slowly, you can imagine your brain like an ocean at night. Some waves move quickly near the surface. Others roll slowly and deeply below. The tide shifts without hurry, guided by internal clocks and quiet chemistry. You do not need to hold on to any of this. Understanding can fade as easily as a wave dissolving into shore. In the next part of our journey, we will step more closely into one particular state, the quiet borderland where wakefulness loosens and dreams begin to form, where the mind turns inward and images gather softly in the dark. Inside the quiet architecture of your brain, beyond the steady rhythm of waves and the branching forest of neurons, there is another kind of movement. It is softer to describe, yet deeply familiar to you. It is the movement of feeling. Emotions rise and fall across your inner world like weather patterns across a wide sky. Sometimes they pass quickly, like a light breeze. Sometimes they linger, like a long afternoon of rain. And beneath each of them, there is gentle and measurable activity unfolding in the living tissue of your brain. Deep within the temporal lobes rests a small almond-shaped structure called the amygdala. Though modest in size, it plays a central role in detecting emotional significance. When you encounter something meaningful, whether pleasant or unsettling, the amygdala becomes active. It does not create fear or joy on its own. Rather, it helps evaluate what matters. It acts as a kind of watchful sentinel, scanning incoming information for relevance. If you hear a sudden sound in the dark, signals travel quickly from your ears toward this region. The amygdala can respond before your conscious mind fully interprets what the sound is. This rapid pathway allows your body to prepare, just in case. Your heart rate may increase slightly. Muscles may tighten. It is a protective system shaped over countless generations, designed to keep you safe. Yet the amygdala does not work alone. Just behind your forehead lies the prefrontal cortex, a region associated with reflection, planning, and regulation. When emotion stirs, the prefrontal cortex engages in a quiet conversation with deeper structures. It assesses context. It weighs possibilities. It can soften or reinterpret an initial reaction. If the sound you heard turns out to be nothing more than wind against a window, the prefrontal cortex helps calm the body's response. This dialogue between regions allows emotion to be both immediate and flexible. You are not locked into your first reaction. Neural pathways allow adjustment, reframing, and understanding. With practice and experience, these regulatory circuits can strengthen. That is why reflection, mindfulness, and even simple pause can gradually reshape emotional responses over time. Another structure woven into this network is the hippocampus, involved in memory formation. It provides context to emotional experiences. When you feel something strongly, the hippocampus helps anchor that feeling to a particular place and time. This is why certain memories carry vivid emotional color. The amygdala and hippocampus interact closely, ensuring that meaningful events are remembered with clarity. Chemical messengers also play a role in shaping emotional tone. Neurotransmitters such as serotonin, dopamine, and norepinephrine influence mood and motivation. Their levels fluctuate naturally throughout the day, affected by sleep, light exposure, social interaction, and even gentle physical movement. These fluctuations are not signs of instability. They are part of a dynamic system responding to your environment. Hormones released by the body further contribute to emotional experience. When stress arises, the adrenal glands release cortisol, preparing you to respond to challenge. In moderate amounts, this hormone supports alertness and adaptation. But when stress is prolonged, elevated cortisol can affect memory and mood. The brain monitors these levels through feedback loops, adjusting production to maintain balance whenever possible. There is also a network known as the limbic system, which includes the amygdala, hippocampus, and other interconnected regions. This system integrates emotion, memory, and motivation. It links bodily sensations to feeling states. A racing heart, a warm flush, a tightening in the chest. These physical cues are interpreted in light of context and experience, becoming part of the emotional landscape. You may notice that emotions often feel embodied. That is because the brain and body are in constant communication. Signals travel along the vagus nerve, connecting internal organs to the brainstem. Changes in breathing, heart rate, and digestion influence how you feel. Slow, steady breathing can activate calming pathways, sending signals upward that support relaxation. There is nothing you need to force. Even small shifts can gently guide the system toward ease. Interestingly, the same neural circuits involved in personal emotion also respond when you observe emotion in others. Mirror neuron systems activate when you witness someone smile or frown, allowing a kind of shared understanding. This capacity underlies empathy, enabling connection without conscious effort. Social bonds influence emotional stability, shaping neural pathways over time. Emotions are not fixed traits stored in isolated pockets of the brain. Now, they are patterns of activity emerging from networks interacting moment by moment. Fear, joy, sadness, contentment, each corresponds to particular configurations of firing neurons, chemical signals, and bodily responses. These patterns can overlap and blend, creating subtle shades of experience. As you lie here now, perhaps feeling calm or gently reflective, certain networks are more active while others rest. The amygdala is not on high alert. The prefrontal cortex is not straining to solve urgent problems. Instead, activity may be balanced, steady. If a thought drifts in that carries emotion, it passes through these circuits and settles again. There is nothing wrong with having emotions. They are not disruptions in the system. They are information, signals that help you navigate the world. Over time, the brain learns which responses are helpful and which can soften. This learning occurs gradually, through repeated experience and quiet reinforcement. Sleep also influences emotional balance. During certain sleep stages, especially those involving dreaming, emotional memories are processed and integrated. Activity patterns in the amygdala and prefrontal cortex shift, allowing experiences to be revisited in altered form. By morning, intense feelings may feel less sharp. The brain has gently reorganized them overnight, so your emotional tides are not random storms. They are guided by networks shaped through evolution, experience, and daily rhythm. They rise, they crest, and they fall. You do not need to hold on to any particular feeling right now. You can let it drift like a cloud across an evening sky. As we continue this quiet exploration, you may begin to wonder how the brain at rest continues its subtle work. How even in stillness, patterns of activity weave together thought and memory, preparing the ground for dreams yet to come. When you think of rest, you might imagine silence, a dimmed room, a body stretched comfortably beneath a blanket, muscles softening, breath slowing. And yet, inside your head, rest does not mean absence. It does not mean shutdown. Even now, as you lie still and allow your thoughts to drift, your brain remains quietly active, not in urgency, but in gentle continuity. There is a network of regions that becomes especially engaged when you are not focused on the outside world. Scientists call it the default mode network. It includes parts of the medial prefrontal cortex, the posterior cingulate cortex, and areas along the parietal lobes. These regions communicate more strongly with one another when your attention is not fixed on a specific task. When you are daydreaming, remembering, reflecting, imagining, this network does not shout. It hums softly in the background, like distant conversation in another room. When you close your eyes and let your mind wander, it begins to weave together fragments of memory, bits of future possibility, small pieces of identity. It is active when you think about yourself, when you recall a past moment, when you consider someone else's perspective. In these quiet states, the brain turns inward. You may notice that when you are not busy solving a problem or responding to a demand, your thoughts drift naturally. One image leads to another. A memory connects to a feeling. A question appears and fades. This wandering is not a flaw. It is a built-in feature of how the brain organizes experience. During these moments, different regions exchange information, strengthening certain connections and gently revising others. Even during wakeful rest, the brain consumes a steady supply of energy. Blood flows continuously through its intricate vessels. Oxygen and glucose nourish neurons as they maintain electrical balance and communicate across synapses. In fact, much of the brain's energy use occurs during these baseline states, rather than during intense bursts of activity. Rest is not inactivity. It is maintenance, integration, quiet coordination. There is nothing you need to force here. If your thoughts move slowly, that is natural. If they move quickly and then settle, that too is natural. The brain shifts fluidly between focused attention and open wandering. These states complement each other. Periods of concentrated effort are balanced by periods of internal reflection. Just as muscles require recovery after exertion, cognitive systems benefit from gentle mental space. Research suggests that during rest, the brain replays patterns of activity associated with recent experiences. Neurons that fired together earlier in the day may activate again in subtle sequences, reinforcing connections. This replay helps consolidate memory, strengthening learning without conscious effort. You do not feel it happening. It unfolds beneath awareness, like gardeners tending soil overnight. At the same time, the brain evaluates information gathered throughout the day. Emotional tones are reconsidered. Decisions are weighed unconsciously. The default mode network interacts with limbic structures and memory centers, forming a kind of internal dialogue. It is not a dialogue in words, but in patterns of activity. Electrical signals rising and falling in coordinated waves. If you pause now and notice your breathing, you may sense a quiet spaciousness. That spaciousness corresponds to shifting neural patterns. When attention turns inward, sensory regions become less dominant. Instead, networks involved in self-referential thought become more engaged. This is why rest often brings reflection. You may find yourself revisiting past conversations or imagining future possibilities without deliberate intention. Interestingly, meditation practices often aim to alter activity in the default mode network. Studies show that experienced meditators can reduce activity in these regions, especially when focusing attention on the present moment. This reduction is associated with decreased self-referential thinking and increased feelings of calm. It is not that the network disappears. Rather, its activity is modulated, becoming less dominant. Sleep itself begins with subtle shifts in these resting patterns. As you grow drowsy, communication between certain regions changes. The thalamus, which acts as a relay for sensory information, begins to reduce its responsiveness to external input. This allows the brain to turn more fully inward. External sounds may still be registered, but their significance fades. During early stages of sleep, the default mode network gradually quiets, on giving way to different patterns of coordination. Yet even then, internal processing continues. Memory consolidation, emotional integration, and metabolic regulation unfold in carefully timed sequences. Rest evolves into restoration. There is comfort in knowing that your brain works gently on your behalf, even when you are doing nothing at all, even when you feel unproductive, even when you lie quietly in the dark. The absence of outward action does not mean stagnation. Beneath the surface are billions of neurons exchange signals, refine connections, and maintain balance. You might imagine your mind as a calm lake at night. On the surface, there is little movement, but beneath, subtle currents flow. Tiny organisms drift. The ecosystem continues its quiet cycles. Stillness and activity coexist. If thoughts arise now, you can let them pass without effort. They are part of the resting brain's natural rhythm. There is no need to hold on to any particular idea. The network will shift on its own. Attention may narrow, then widen. Images may form, then dissolve. You are not required to steer this process. Over time, habitual patterns of thought can shape resting activity. Repeated worries may strengthen certain pathways. Repeated gratitude may strengthen others. The brain adapts to what it practices. Yet this adaptation is gradual, unfolding across months and years. It is not a fragile system that changes overnight. It is resilient, flexible, responsive. As evening deepens and external light fades, signals from your eyes help align internal clocks. Hormonal changes support relaxation. Neural activity slows in particular regions. The resting brain prepares for the transition into deeper sleep, but it does so at its own pace, guided by rhythms older than conscious awareness. You are allowed to rest here. There is nothing to solve in this moment. The quiet hum within your head is enough. It carries forward memories, shapes identity, prepares dreams. Even silence has structure. And soon, as the brain's resting patterns soften further and sensory gates close more gently, you may begin to cross into that subtle threshold where imagination loosens from waking logic, and the first fragments of dreams begin to gather in the dark. As wakefulness loosens its gentle grip and your thoughts begin to drift without effort, the brain shifts into a different mode of being. Sleep does not arrive all at once. It unfolds gradually, like twilight deepening into night. The rhythms you explored earlier begin to slow, and communication between certain regions softens. The world outside grows quieter, and the world within grows more vivid. In the earliest stage of sleep, your awareness thins but does not disappear. Muscles relax. Breathing becomes slower and more regular. Brain activity shifts towards slower waves, and you may experience fleeting images or sensations that feel almost dreamlike. These are sometimes called hypnagogic images, brief fragments of thought that float through consciousness as you cross the threshold from waking into sleep. You do not need to hold on to them. They pass naturally. As sleep deepens, the body settles further. Heart rate slows, temperature drops slightly. Growth hormone is released, supporting repair and renewal. Meanwhile, neural activity changes in coordinated ways. Certain regions reduce responsiveness to external input. The thalamus, which normally relays sensory information to the cortex, begins to filter signals more selectively. Sounds may still reach your ears, but their significance fades. The brain turns inward. After a period of deeper non-REM sleep, a different state begins to emerge. Brain activity becomes more varied and dynamic. Rapid eye movements flicker beneath closed lids. Muscles remain largely relaxed, prevented from acting out imagined movements by signals from the brainstem. This stage is known as REM sleep, and it is here that dreaming often becomes most vivid. During REM sleep, parts of the brain associated with visual imagery and emotion become more active. The occipital regions light up in patterns similar to those seen during waking vision. The amygdala, involved in emotional processing, shows increased activity. Yet the prefrontal cortex, responsible for logical reasoning and self-monitoring, becomes less active. This shift allows dreams to unfold with unusual narratives, blending memory and imagination without strict adherence to waking logic. You become both storyteller and audience. Neural networks draw upon stored memories, fragments of recent experience, and emotional tones. These elements combine in novel ways. A familiar place may merge with a different time. A known face may appear in an unexpected setting. The brain is not trying to confuse you. It is reorganizing information, integrating experience into broader patterns. REM sleep occurs in cycles throughout the night, typically every 90 minutes or so. Early cycles may contain shorter periods of REM, while later cycles can extend longer. As morning approaches, REM stages often become more prolonged. This cycling between non-REM and REM sleep reflects the brain's coordinated timing, guided by internal clocks and chemical changes. Neurotransmitter levels shift dramatically during these stages. During REM sleep, certain chemicals such as acetylcholine become more active, supporting cortical activation. Others, like serotonin and norepinephrine, decrease. These changes create a distinct chemical environment, shaping the tone of dreaming. You are not required to manage this balance. It unfolds automatically. Dreaming may serve multiple functions. Research suggests it contributes to emotional processing, allowing the brain to revisit experiences in a context where stress responses are reduced. The amygdala remains active, but stress hormones such as cortisol are lower during much of the night. This combination may help soften emotional intensity over time. Memories are woven into broader narratives, losing some of their sharp edges. At the same time, memory consolidation continues. Patterns of neural activity associated with learning are replayed during both REM and non-REM sleep. Connections strengthen or weaken based on prior use. Synapses adjust their sensitivity. The brain refines its internal maps without conscious effort. Learning does not pause when you sleep. It quietly reorganizes. The muscle relaxation that accompanies REM sleep is not accidental. Signals from the pons in the brainstem inhibit motor neurons in the spinal cord, preventing large muscle movements. This protective mechanism ensures that dream actions do not translate into physical movement. Though your eyes may dart gently beneath your lids, your body remains safely still. Breathing during REM sleep can become more irregular, reflecting the variability of dream content. Heart rate may fluctuate slightly, yet these changes remain within healthy ranges, guided by autonomic regulation. The body and brain move through these shifts with quiet coordination. Not all dreams are remembered. Memory of dreams depends partly on whether you awaken during or shortly after REM sleep. If you wake gently during a dream phase, fragments may linger in your awareness. If you continue sleeping, they often fade. This fading is natural. The purpose of dreaming is not necessarily to provide vivid recollection, but to support internal processing. Even in deeper non-REM sleep, when dreams are less vivid, important processes unfold. Slow waves dominate, and the brain engages in large-scale synchronization. This stage supports physical restoration and memory stabilization. Cerebrospinal fluid circulates more freely, assisting in metabolic cleanup. The sleeping brain remains active in ways that sustain long-term health. You may notice that sleep is not a single state, but a carefully orchestrated sequence. Different networks activate and quiet in turn. Hormones rise and fall. Neural patterns shift with remarkable precision. All of this occurs without conscious control. There is nothing you need to do to make it happen. As you rest now, perhaps drifting closer to that threshold, you can imagine the brain preparing its nightly theater. Lights dim in some regions and brighten in others. Memories take their places like actors waiting behind a curtain. Emotional tones settle into softer hues. The audience and the storyteller are one and the same. Sleep is not emptiness. It is transformation. It is the brain turning inward to weave experience into coherence. Each night offers this opportunity for quiet integration. Each morning emerges from cycles completed in darkness. And as dreams gather gently at the edge of awareness, you may begin to wonder how the brain reshapes itself through learning and experience. How these nightly rehearsals influence the very pathways that form who you are becoming. If you imagine the brain as a landscape, then learning is the slow shaping of its pathways. Nothing dramatic, no sudden cliffs rising overnight. Instead, change happens the way a footpath forms across an open field. At first, the grass stands tall and unmarked. Then someone walks across it once. A faint trace appears. With each return, the path becomes clearer, easier to follow. Over time, it feels natural to step there. The brain changes in much the same way. This ability to change is called plasticity. It does not mean the brain is soft like clay waiting to be molded all at once. It means that connections between neurons can strengthen, weaken, form, or dissolve in response to experience. When two neurons are active at the same time repeatedly, the synapse between them becomes more efficient. Signals pass more easily. The pathway becomes preferred. You experience this when you practice something new. Perhaps you learn a language, an instrument, or even a simple route through a city. At first, effort is required. Signals move through unfamiliar circuits. Gradually, repetition refines these pathways. Myelin, the insulating layer around certain axons, can increase with repeated use, allowing signals to travel more quickly. What once felt awkward begins to feel smooth. This change does not occur only in childhood. While early life is a period of especially rapid growth and connection, the adult brain retains the capacity to adapt. New synapses can form. Existing ones can adjust their sensitivity. Even in later years, learning continues to reshape neural networks. The pace may be slower, but the potential remains. Plasticity also involves pruning. Connections that are rarely used may weaken over time. This is not loss for its own sake. It is refinement. By reducing less active pathways, the brain maintains efficiency, conserving energy and focusing resources on patterns that matter most. Just as a gardener trims excess branches to allow healthier growth, the brain reshapes itself to support clarity. Emotion influences plasticity as well. Experiences that carry strong emotional weight tend to leave deeper traces. The amygdala interacts with memory systems, enhancing consolidation of significant events. This is why moments of joy, fear, or surprise often feel especially vivid in recall. Chemical signals released during emotional arousal strengthen synaptic change, anchoring the memory more firmly. Sleep plays a vital role in this process. During certain stages, patterns of neural activity associated with recent learning are replayed. These reactivations strengthen connections, transferring information from temporary storage toward more stable networks. The brain practices quietly in the dark, refining what it encountered during the day. You are not aware of this rehearsal. It unfolds beneath the surface of consciousness. Even subtle experiences can shape pathways. Repeated thoughts, habits of attention, small daily routines, all influence synaptic strength. If you frequently focus on gratitude, networks associated with positive appraisal may strengthen. If you repeatedly rehearse worry, those circuits may become more easily activated. the brain adapts to what it practices. This adaptation is gradual, built through consistency rather than force. There is no need to rush this change. Neural remodeling takes time. Proteins are synthesized. Receptors are inserted or removed at synapses. Structural changes occur in dendritic spines, the tiny protrusions that receive signals. These biological adjustments require energy and patience. Just as a tree does not grow visibly taller in a single night, neural change accumulates slowly. Our injury to certain areas of the brain can also reveal plasticity. When one region is damaged, neighboring areas sometimes take on additional roles. Rehabilitation and practice can help recruit alternative pathways. The brain seeks balance and function where possible, redistributing tasks across its networks. This resilience reflects the flexible architecture you have already explored. Environment influences plasticity too. Enriched surroundings, social interaction, and varied experiences can stimulate synaptic growth. Isolation or prolonged stress may have the opposite effect. Hormones like cortisol, elevated during chronic stress, can affect the hippocampus and other regions involved in memory. Yet even then, supportive conditions and healthy habits can promote recovery over time. Learning is not only about acquiring new information, it is about reshaping internal maps. When you encounter something unfamiliar, prediction systems within the brain adjust. The cortex constantly generates expectations about incoming sensory input. When reality differs from expectation, signals update the model. This predictive coding allows adaptation without constant conscious effort. You might notice that some skills, once learned, feel automatic: riding a bicycle, typing on a keyboard, speaking a native language. These actions rely on deeply embedded neural circuits. Repetition has strengthened those pathways so thoroughly that they operate with minimal attention. Plasticity has allowed the brain to streamline them. There is comfort in knowing that change remains possible. Even small adjustments, repeated gently, can alter the landscape over time. A new habit practiced consistently can carve a fresh path through the field. A perspective reconsidered repeatedly can soften an old reaction. Nothing in the brain is permanently frozen. As you rest now, and perhaps reflecting softly, your brain continues its quiet remodeling. Synapses shift subtly in response to what you have experienced today. Pathways strengthen or weaken according to patterns of use. You do not need to direct this process. It is part of being alive. Sleep reminder here. If your thoughts begin to slow, that is welcome. Plasticity does not require alert concentration in this moment. It is happening even as you drift. The gentle repetition of breath, the calm tone of voice, and the steady rhythm of rest. All of these contribute small signals that shape neural patterns over time. And as learning continues to reshape the pathways within you, it naturally connects to the ways you perceive the world through your senses. How light, sound, touch, and scent enter this adaptable network and become part of who you are becoming. Before a thought forms, before a memory awakens, there is sensation. The world reaches you through small, patient doorways. Light touches the eyes. Air vibrates against the ears. Fabric brushes the skin. Molecules drift into the nose. Flavor settles across the tongue. These signals arrive quietly, and your brain receives them without ceremony, translating physical events into lived experience. Sight begins when particles of light enter the curved surface of the eye. They pass through the cornea and lens, which gently focus them onto the retina at the back. There, specialized cells called photoreceptors respond to brightness and color. Rod cells are sensitive in low light, helping you see in dim conditions. Cone cells detect color and fine detail. When light strikes these cells, chemical changes trigger electrical signals that travel along the optic nerve toward the brain. These signals reach the occipital cortex, where patterns are analyzed, edges are detected, motion is tracked, depth is inferred. You do not experience the intermediate steps. You simply see a room, a shadow, a shape. Yet beneath that seamless image, countless neurons are coordinating in precise timing. The brain does not passively record the world. It actively constructs perception based on incoming data and prior knowledge. Sound follows a different path, but a similar principle. Vibrations in the air enter the ear canal and strike the eardrum, causing it to move. Tiny bones in the middle ear amplify these vibrations, transmitting them to the cochlea, a fluid-filled spiral in the inner ear. Inside the cochlea, hair cells bend in response to fluid motion, converting mechanical energy into electrical signals. These signals travel along the auditory nerve to the temporal lobes. In the auditory cortex, frequency, intensity, and timing are decoded. Speech is distinguished from noise. Melody is separated from rhythm. The brain identifies patterns and assigns meaning. When you hear a familiar voice, recognition occurs in an instant. Emotional tone is processed alongside linguistic content, engaging both auditory and limbic regions. Hearing is not just detection, it is interpretation. Touch begins in receptors scattered throughout your skin. Some respond to pressure, others to temperature, others to vibration or pain. When activated, they send signals along sensory nerves toward the spinal cord and up to the brain. The parietal lobe contains a map of the body's surface, known as the somatosensory cortex. Different regions correspond to different body parts. Areas requiring fine sensitivity, such as the fingertips and lips, occupy more cortical space. When you feel the softness of a pillow or the coolness of night air, this map becomes active. Signals arrive and are integrated into a coherent sensation. At the same time, emotional centers may respond, adding comfort or alertness. Even simple touch carries layers of meaning shaped by context and memory. Smell travels directly from the nose to deeper brain structures. The odor molecules bind to receptors in the nasal cavity, triggering signals that pass to the olfactory bulb. From there, information moves to regions including the amygdala and hippocampus. This close connection to emotion and memory explains why certain scents can evoke vivid recollections. A faint fragrance may bring back an entire scene, complete with feeling. Taste works through receptors on the tongue that detect basic qualities such as sweetness, saltiness, sourness, bitterness, and savory richness. Signals from these receptors travel to the brainstem and onward to the gustatory cortex. Yet flavor is not taste alone. It combines smell, texture, temperature, and even visual expectation. The brain integrates all these inputs to create the experience of eating. You may notice that perception is not a direct copy of reality. The brain constantly predicts what it expects to encounter. When sensory input matches expectation, perception feels stable. When it differs, the brain adjusts its internal model. This predictive process allows efficient interpretation, but can also lead to illusions, when context misleads expectation. Sensory information does not enter consciousness all at once. The thalamus acts as a relay station for most senses, directing signals to appropriate cortical areas. It filters and prioritizes input, preventing overload. Without this filtering, every minor stimulus would demand attention. Instead, you are able to focus selectively, guided by both external cues and internal goals. Vision and hearing often dominate awareness, but the body continuously monitors internal states as well. Receptors detect blood pressure, oxygen levels, and the stretch of organs. These signals travel to the brainstem and insular cortex, contributing to a sense of internal balance. This internal perception, called interoception, informs emotional tone and bodily awareness. As you rest now, many of your senses are quieted. Eyes closed, visual input diminishes. The room grows softer in sound. Touch remains as a gentle presence, the weight of blankets, the surface beneath you. Even in this quiet state, signals continue to flow. The brain does not cease interpreting simply because the environment is calm. There is no need to analyze these sensations. They arise and fade naturally. The brain organizes them into experience without your conscious effort. You are not required to supervise the process. It unfolds continuously, a seamless dance between external input and internal construction. Over time, sensory experiences shape neural pathways. Repeated exposure to certain patterns refines perception. Musicians develop heightened sensitivity to pitch. Artists perceive subtle shifts in color. Chefs distinguish delicate flavor notes. Practice strengthens relevant circuits, illustrating once again the brain's capacity for change. And as sensory signals blend into memory and emotion, they also connect you to others. Our shared sights, sounds, and tastes form common ground. Perception becomes part of social understanding. In the next gentle step of our journey, we may explore how this brain, shaped by sensation, learns to connect with other minds. How empathy and shared experience arise within its quiet networks. From your very first breath, you are not alone in your wiring. The human brain develops in the presence of others, shaped by faces, voices, touch, and shared attention. Even before language forms, your nervous system responds to rhythm and expression. A caregiver's smile activates networks in your visual cortex. A soothing voice steadies your heart rate. Connection is not an added feature layered onto an independent mind. It is woven into the architecture itself. When you look at another person's face, specialized regions in the temporal lobe become active. The fusiform gyrus helps you recognize familiar features, distinguishing one individual from another. Subtle shifts in expression are detected by networks that interpret emotion. A raised eyebrow, a softened gaze, a tightening around the mouth. These details are processed rapidly, often before you consciously reflect on them. Social perception is swift and intuitive. Deeper within the brain, structures such as the amygdala evaluate the emotional significance of these cues. It does not simply respond to threats, it responds to relevance. A friendly smile may trigger warmth. A stern expression may prompt caution. These reactions are not judgments, but adaptations, helping you navigate complex social landscapes. There are also neurons that activate both when you perform an action and when you observe someone else performing the same action. Often referred to as mirror neurons, These cells were first identified in studies of primates and are believed to exist in humans as well. When you watch someone grasp a cup, certain motor regions in your own brain reflect that movement pattern. This mirroring supports understanding through internal simulation. You sense, in a subtle way, what it might feel like to perform the action yourself. Empathy grows from these shared activations. When you see someone in distress, areas involved in processing your own emotional pain can become active. The anterior cingulate cortex and insula respond to both personal discomfort and observed suffering. This overlap allows you to resonate with another's state. It does not mean you feel exactly what they feel, but it creates a bridge of understanding. Language further deepens this connection. Regions in the left hemisphere coordinate the production and comprehension of speech. When you listen to someone speak, auditory areas decode sounds, while language networks assign meaning. At the same time, social cognition regions consider intention and context. Communication is not merely exchange of words, it is alignment of minds. The brain also contains circuits dedicated to understanding perspective. The medial prefrontal cortex and temporoparietal junction become active when you consider another person's thoughts or beliefs. This capacity, sometimes called theory of mind, allows you to imagine experiences beyond your own. It supports cooperation, teaching, storytelling. Without it, social life would be fragmented. A connection influences biology as well. Positive social interaction can stimulate the release of oxytocin, a hormone associated with bonding and trust. Oxytocin interacts with neural circuits involved in reward and stress regulation, supporting feelings of safety. When you feel understood, stress responses often diminish. Heart rate steadies, muscles soften. The body responds to social cues as much as to physical ones. Chronic isolation, by contrast, can affect neural pathways related to mood and cognition. Studies show that prolonged loneliness may alter activity in regions involved in emotional regulation. The brain, accustomed to connection, adapts when it is absent. Yet, just as plasticity allows change through repetition, renewed connection can reshape these patterns over time. Even subtle synchrony between people reflects neural coordination. When individuals engage in conversation or shared activity, patterns of brain activity can become aligned. Research using hyperscanning techniques suggests that speakers and listeners sometimes exhibit synchronized neural rhythms during meaningful exchange. It is as though two separate systems briefly move in harmony. You may notice how easily laughter spreads within a group. Hearing someone laugh can activate motor regions associated with producing laughter, making you more likely to join in. Emotional contagion operates through shared circuitry. The brain reflects others like calm water reflecting moonlight, capturing fragments of expression and returning them in kind. There is nothing you need to force in this understanding. Your brain is already equipped for connection. Even in solitude, memories of relationships activate social networks. Imagining a loved one's face can engage many of the same regions involved in direct interaction. The social brain remains active, weaving bonds through memory and imagination. Developmentally, these circuits strengthen through experience. Infants learn to track gaze direction, follow pointing gestures, and respond to vocal tone. Repeated interaction shapes synaptic connections supporting social cognition. Over time, cultural norms and personal history influence how these networks interpret signals. Social understanding becomes both biological and contextual. As you rest now, perhaps recalling a familiar face or shared moment, certain pathways quietly activate. Now, you do not need to analyze them. The resonance is gentle. Connection does not require constant contact. It lives within neural patterns shaped across years. Sleep reminder here. If thoughts of others drift through your mind, let them pass softly. The brain's social networks continue their subtle dialogue, integrating memory and emotion. You are wired for relationship, yet you are also safe in this quiet space. There is nothing to perform, nothing to prove. And as connection intertwines with perception and learning, it naturally leads toward another quiet question: how attention moves through these networks, narrowing and widening like a beam of light across the landscape of your mind. Attention is often described as a spotlight, but perhaps it is softer than that. Imagine instead a warm lantern carried through a quiet field at night. Wherever you turn it, certain shapes become clearer, colors sharpen, edges stand out. Beyond the glow, other details remain present but less defined. This is how your brain manages the steady stream of information arriving each moment. It does not process everything equally. It selects, enhances, and lets the rest settle into the background. Within your brain, attention arises from coordinated activity between several regions. The prefrontal cortex, resting just behind your forehead, plays a guiding role. It helps decide what deserves focus based on goals and context. Now, the parietal lobes assist in directing that focus towards specific locations or features. Together, these regions form networks that shift resources toward what matters right now. When you concentrate on a task, neurons in relevant sensory areas increase their activity. Signals representing the chosen object or thought become stronger, while competing signals are gently suppressed. This selective enhancement allows clarity. If you are reading, visual regions sharpen the lines of text. If you are listening closely, auditory areas heighten sensitivity to subtle sounds. Yet attention is not fixed. It moves. Even when you try to hold it steady, it may drift. This wandering is not weakness. It is part of how the brain balances focus with exploration. Networks associated with directed attention alternate with networks linked to internal reflection. The brain oscillates between engagement with the external world and movement within its own landscape. There is a network sometimes called the dorsal attention system, which activates when you deliberately focus on something specific. It supports goal-directed concentration. There is also a ventral attention system, which responds to unexpected stimuli, drawing your awareness towards sudden changes. This system helps you notice important events, like a new sound in a quiet room. Both networks cooperate, ensuring flexibility. When attention shifts inward, now the default mode network becomes more active. Thoughts drift across memory and imagination. You may find yourself replaying a conversation or anticipating a future event. This mental movement often happens without conscious decision. It is as natural as breathing. The brain explores possibilities, revisits experiences, and integrates emotion during these wandering moments. You might notice that prolonged concentration requires energy. Maintaining focus engages sustained neural coordination. Neurotransmitters, such as dopamine and norepinephrine, modulate alertness and signal-to-noise ratio within circuits. When levels are balanced, focus feels steady. When they fluctuate, attention may feel scattered or fatigued. This variability is biological, not moral. There is nothing wrong with distraction. The brain evolved to scan the environment for change. In early human history, noticing subtle shifts in sound or movement could signal opportunity or danger. Even now, your nervous system remains attuned to novelty. A sudden vibration or flicker of light can capture your awareness automatically. This reflex protects you, even if it sometimes interrupts your train of thought. Training attention gently strengthens related pathways. Practices such as meditation encourage returning awareness to a chosen anchor, like the breath. Each time you notice distraction and redirect focus, circuits supporting self-regulation become more refined. This process resembles the formation of a footpath across grass. Repetition gradually increases ease. However, constant strain is unnecessary. The brain benefits from alternating periods of focus and rest. Creative insights often arise when attention relaxes. During mind-wandering, distant associations may connect in new ways. Without the constraints of directed control, the brain can combine ideas freely. Many discoveries and artistic inspirations emerge from these quiet intervals. Attention also shapes perception. When you focus on one element of a scene, other elements may fade from awareness. This phenomenon reflects limited processing capacity. The brain prioritizes certain inputs while filtering others. It is not that unseen details disappear entirely. They remain accessible if attention shifts toward them. You may recall moments when you drove a familiar route and later realized you barely remember the journey. In such cases, well-learned behaviors rely on automatic circuits, freeing attention for internal thought. This automaticity reflects efficient neural encoding. The brain allocates resources where novelty or complexity demands them. As you lie here now, your attention may narrow toward the sound of this voice, or it may drift outward toward subtle sensations in the room, or it may turn inward toward a passing memory. All of these movements are valid. There is no requirement to hold focus perfectly. Even the act of noticing wandering is itself a form of attention. Neuroscientists study these dynamics using imaging techniques that reveal shifting patterns of blood flow across regions. When tasks demand concentration, activity rises in frontal and parietal areas. When the mind rests or reflects, midline structures grow more active. These fluctuations are rhythmic, part of the brain's ongoing choreography. Fatigue influences attention as well. As sleep pressure builds, signals from the brainstem and hypothalamus alter arousal systems. Maintaining sharp focus becomes more difficult. Micro-lapses may occur. This is not failure. It is biology signaling the need for rest. The brain protects itself by gradually softening vigilance. There is comfort in understanding that your wandering mind is not broken. It is adaptive. It explores, it scans, it integrates. Focus and drift are two expressions of the same flexible system. Like wind moving through tall grass, thought bends and rises again, guided by currents you do not have to control. If you choose to rest your awareness gently on your breathing, that is enough. If it moves elsewhere, that is enough too. The brain's attention networks will continue their quiet balancing act. Over time, repeated gentle redirection can shape these circuits. But tonight, there is nothing you must practice. And as attention gradually softens and internal reflection grows more spacious, it opens the door to another quiet faculty, the capacity to imagine, to create, to allow new ideas to bloom softly within the adaptable landscape of your mind. Creativity rarely arrives as a thunderclap. More often, it unfolds like a slow sunrise, where light gradually reveals shapes that were already present in shadow. Inside your brain, imagination is not confined to a single hidden chamber. It emerges from the quiet collaboration of many regions, each contributing its own subtle influence. When these regions coordinate in new ways, ideas begin to bloom. You might think of creativity as the weaving together of distant threads. Memories stored in the temporal lobes, sensory impressions mapped across the cortex, emotional tones shaped by deeper structures. All of these elements can combine in novel arrangements. The brain does not invent from nothing. It rearranges, recombines, and refines what it has already experienced. Research suggests that creative thinking often involves interaction between the default mode network and executive control systems. The default mode network supports internal reflection, memory retrieval, and spontaneous association. The executive network, centered in parts of the prefrontal cortex, helps evaluate and shape these associations into coherent forms. When imagination flows freely, these systems do not compete. They alternate and cooperate. In moments of relaxed reflection, the default mode network becomes active, allowing thoughts to drift across memory and possibility. Associations arise without strict direction. An image from childhood may blend with a recent conversation. A melody may connect with a color. This spontaneous linking reflects the brain's vast interconnectedness. Neurons that once fired together may activate again, even in new contexts. Yet creativity is not only wandering. At some point, evaluation enters. The prefrontal cortex assesses which ideas feel promising and which fade gently away. This evaluation does not need to be harsh. It is a quiet filtering process, selecting patterns that align with goals or meaning. The balance between openness and guidance shapes creative output. Dopamine, a neurotransmitter involved in reward and motivation, plays a role in this process. When you generate a novel idea that feels satisfying, dopamine pathways may become active, reinforcing that line of thought. This subtle chemical signal encourages exploration and persistence. Curiosity itself is supported by these reward circuits, gently guiding attention toward novelty. Imagination also draws upon sensory regions. When you picture a landscape with your eyes closed, visual areas in the occipital cortex become active. Though no external light enters, the brain simulates perception internally. Similarly, when you imagine a melody, auditory regions respond. The mind can rehearse experiences without external input, activating many of the same circuits used in real perception. Children often display vivid imaginative play, reflecting flexible neural connections and abundant exploratory behavior. In adulthood, creativity remains possible, though shaped by accumulated experience and refined networks. Practice strengthens pathways involved in artistic or problem-solving skills. Repetition does not diminish originality. It deepens fluency, allowing ideas to move more freely. Interestingly, Moments of insight frequently occur after periods of incubation. You may struggle with a question, set it aside, and later find an answer appearing unexpectedly. During the break, background networks continue processing. The brain reorganizes information without conscious effort. When conditions align, a solution rises gently into awareness. Sleep contributes to this incubation. During certain stages, memory fragments are replayed and recombined. Associations not obvious during wakefulness may form overnight. Studies show that people sometimes solve problems more effectively after sleep, particularly when tasks require integration of distant ideas. The dreaming mind can explore combinations beyond strict logic. Creativity is not limited to art or invention. Everyday problem-solving, conversation, and adaptation all require imaginative flexibility. When you consider another person's perspective, you simulate their experience internally. When you plan for tomorrow, you construct possible futures in your mind. These acts draw upon the same neural foundations as poetry or painting. Stress can narrow creative range by heightening activity in circuits focused on immediate survival. When the amygdala signals threat, resources shift toward vigilance. In calmer states, the brain allows broader exploration. This is one reason relaxation often supports imaginative thought. When you feel safe, networks can wander more freely. There is nothing you need to force tonight. Creativity does not demand performance in this moment. Even as you rest, networks within your brain remain capable of subtle recombination. Ideas may drift softly through your awareness or settle quietly beneath it. Both states are natural. Sleep reminder here. If your thoughts grow hazy, that is welcome. Imagination does not require sharp clarity. In fact, the borderland between waking and sleep often brings gentle imagery. Shapes blend, scenes shift. These transitions reflect the brain's ongoing interplay between memory, perception, and emotion. Over time, repeated engagement in creative activity can strengthen connectivity between distant regions. White matter pathways may become more efficient, supporting communication across networks. Experience sculpts structure. Structure influences possibility. The cycle continues, patient and continuous. You might picture your mind as a vast sky before dawn. At first, only darkness. Then faint hues begin to appear. Gradually, colors deepen, revealing forms that were always there. Creativity often unfolds this way, not as sudden invention, but as gradual illumination. And as imagination continues its quiet work, shaped by chemistry, memory, and rest, it naturally connects to the subtle biological currents that influence mood and motivation. The gentle chemistry that flows through your brain, guiding calm, energy, and intention. Beneath the electrical whispers of neurons, beneath the slow tides of brainwaves, there is another layer of quiet influence. It does not flash or spark, it flows. Tiny molecules drift across microscopic spaces, carrying signals from one cell to the next. These are neurotransmitters, and though they are invisible to you, they shape the tone of your thoughts, the steadiness of your mood, and the gentle pull of motivation. When one neuron releases a neurotransmitter into the narrow space of a synapse, the molecule crosses the gap and binds to a receptor on the neighboring cell. This binding can increase the likelihood that the next neuron will fire, or it can soften activity, creating balance. Each type of neurotransmitter has its own tendencies, its own rhythm of influence. None acts alone. They form a shifting symphony, adjusting in response to experience, sleep, light, and connection. Dopamine is often described as a chemical of reward, but it is perhaps more accurate to think of it as a signal of significance. When you anticipate something meaningful or notice progress toward a goal, dopamine pathways become active. These pathways originate in deeper regions, such as the ventral tegmental area, and project forward to the prefrontal cortex and other structures. The gentle rise of dopamine can make effort feel worthwhile. It does not create pleasure in isolation. It highlights possibility. Serotonin contributes to a sense of steadiness. It is produced in clusters of neurons in the brainstem and distributed widely throughout the brain. Balanced serotonin activity is associated with mood regulation and a sense of well-being. Light exposure, social interaction, and regular sleep patterns can influence its levels. This is why a calm morning walk or a steady daily rhythm can subtly shift how you feel. The chemistry responds to environment. Another messenger, gamma-aminobutyric acid, often abbreviated as GABA, plays a calming role. It is inhibitory, meaning it reduces the likelihood of neuronal firing. When GABA binds to its receptors, it allows chloride ions to enter the neuron, making it less excitable. This mechanism supports relaxation and helps prevent excessive activity. Many sleep-promoting and anti-anxiety medications enhance GABA signaling, though your brain naturally regulates it as well. Glutamate, by contrast, is the primary excitatory neurotransmitter. It facilitates learning and memory by strengthening synaptic connections. Without glutamate, neural communication would falter. Yet balance is essential. Too much excitation without inhibition can disrupt stability. The brain continuously adjusts levels of glutamate and GABA to maintain equilibrium, like careful weights on a scale. Oxytocin, often associated with bonding, is released during social connection, touch, and trust. It interacts with emotional and reward circuits, supporting feelings of safety and attachment. When you share a warm conversation or receive a comforting gesture, oxytocin may rise gently. This does not guarantee bliss. It simply tilts the internal balance toward connection. Norepinephrine influences alertness and responsiveness. Produced in the locus coeruleus, it prepares the brain to respond to new information. In moderate amounts, it sharpens focus. In excess, it may contribute to anxiety. During sleep, levels of norepinephrine decrease significantly, allowing the brain to enter restorative states. This nightly reduction is part of why rest can feel calming after a busy day. The interplay of these chemicals shifts throughout the day in response to circadian rhythms. Morning light influences hormonal signals that ripple through neurotransmitter systems. As evening approaches, melatonin rises, encouraging the transition toward rest. The chemistry of your brain is not static. It ebbs and flows, guided by internal clocks and external cues. Diet, movement, and stress also influence these systems. Physical activity can increase dopamine and serotonin availability, supporting mood and motivation. Chronic stress, on the other hand, elevates cortisol, which interacts with neurotransmitter pathways and may alter their balance over time. Yet the system remains adaptable. With supportive conditions, equilibrium can gradually return. You do not need to calculate these levels or track each fluctuation. The regulation occurs automatically through feedback loops. When neurotransmitter concentrations rise, receptors may adjust their sensitivity. When levels fall, synthesis and release can increase. This self-regulating nature reflects the brain's commitment to balance. Motivation itself emerges from the integration of chemistry and circuitry. Dopamine signals highlight potential reward. Prefrontal regions evaluate goals. Emotional centers weigh significance. When alignment occurs, action feels natural. When misaligned, effort may feel heavy. This is not a personal failing. It reflects the state of dynamic systems interacting. Calm arises when inhibitory signals balance excitation, when stress hormones recede, when breathing slows and heart rate steadies. You can support this shift gently through posture and breath, but even without deliberate practice, nighttime biology encourages it. As darkness deepens, arousal systems quiet. Neurotransmitter levels adjust. The body prepares for sleep. Sleep reminder here. If your mind begins to soften now, that is a reflection of shifting chemistry. GABA may be exerting greater influence. Norepinephrine may be receding. Melatonin may be rising. You do not feel these molecules directly, but you experience their combined effect as ease. Over years, repeated experiences can shape neurotransmitter pathways. Habits of thought influence receptor sensitivity. Patterns of stress or relaxation leave subtle imprints. Yet plasticity remains. Change is possible, often gradual and steady rather than sudden. The chemistry of calm and motivation is not separate from the networks you have explored. It flows through them, modulating their activity. Electrical signals and chemical messengers cooperate in constant dialogue. One does not exist without the other. As you rest here, imagine these molecules moving like gentle currents beneath still water, adjusting and balancing without noise. There is no need to direct them. They have carried you through countless days and will carry you through the night. And as chemistry continues its quiet work, guiding mood and intention, it invites a final reflection on how the brain adapts across an entire lifetime. How aging and experience shape this remarkable, ever-changing landscape. From your earliest days to the quiet years beyond, your brain is never still in its development. It does not freeze at adulthood, nor does it simply decline in a straight line. Instead, it shifts, adapts, and refines itself across the span of a lifetime. Change is constant, though often subtle. Like the rings inside a tree trunk, each year leaves its trace, not always visible from the outside, but present within the structure. In childhood, the brain grows rapidly. New connections form at astonishing speed as neurons branch and reach toward one another. The young brain produces more synapses than it will ultimately keep. This abundance allows exploration. Through experience, frequently used connections strengthen, while unused ones gradually fade. This process of pruning increases efficiency, helping the brain specialize in response to its environment. As adolescence approaches, further refinements unfold. The prefrontal cortex, involved in planning and self-regulation, continues to mature into early adulthood. Myelination increases along certain pathways, allowing signals to travel more quickly and reliably. Decision-making becomes steadier. Impulses are balanced with reflection. These shifts are gradual, shaped by experience, sleep, social interaction, and learning. In adulthood, The brain maintains remarkable stability while still retaining flexibility. Neural plasticity continues, though at a more measured pace. Learning a new skill, forming new relationships, or navigating unfamiliar challenges can reshape connections. The hippocampus, important for memory, can generate new neurons even in later years, though the rate may vary depending on health and activity. This capacity reflects ongoing renewal. With advancing age, some changes become more noticeable. The processing speed may slow slightly. Certain types of memory, particularly recall of names or recent details, can require more effort. These shifts often relate to gradual alterations in white matter integrity and neurotransmitter levels. Yet, not all abilities decline. Vocabulary and accumulated knowledge often remain stable or even improve. Experience provides a broader framework for interpretation. Wisdom, though difficult to measure precisely, may emerge from decades of integrated experience. Neural networks linking emotion and cognition can become more balanced. Research suggests that older adults sometimes show greater emotional regulation with reduced reactivity to negative stimuli. This may reflect changes in connectivity between the amygdala and prefrontal cortex. Life experience shapes response patterns. Blood flow patterns in the aging brain can also shift. In some cases, when one region shows decreased activity, another may compensate by increasing its engagement. This redistribution reflects adaptability. The brain recruits alternative pathways to support function, demonstrating resilience even when structural changes occur. Lifestyle factors influence how these processes unfold. Regular physical activity supports circulation and can promote neurogenesis in the hippocampus. Social engagement stimulates cognitive networks and may protect against decline. Sleep continues to play a restorative role, supporting memory consolidation and metabolic maintenance. Even modest changes in daily routine can influence long-term brain health. Chronic stress, however, can accelerate certain age-related changes. Elevated cortisol over long periods may affect memory systems and emotional regulation. Yet, reduction of stress through supportive relationships, relaxation practices, and meaningful activity can mitigate these effects. The brain remains responsive to environment throughout life. There is no single timeline for growth or change. Individual variation is wide. Genetics, education, nutrition, health conditions, and lived experience all contribute to how neural patterns evolve. Two people of the same age may show very different cognitive profiles. The brain is not a uniform clock. It is a dynamic ecosystem. Even in later decades, learning remains possible. Studies show that older adults can acquire new languages, master musical instruments, or adapt to new technologies. The pace may differ from youth, but the capacity persists. Synaptic change does not vanish with time. It continues, supported by repetition and engagement. You may notice that memories from earlier life feel vivid, while recent details sometimes blur. This pattern reflects how long-term memory stabilizes through repeated retrieval and integration. Older memories have been rehearsed many times, strengthening their neural pathways. Recent events may still be consolidating. The brain organizes experience across decades, layering new information atop established networks. There is nothing you need to fix in this natural progression. Slower processing does not mean lesser worth. Shifting attention does not mean failure. The brain adjusts its priorities across the lifespan, often placing greater emphasis on emotional meaning and social connection in later years. This rebalancing can support well-being. Sleep reminder here. As you rest now, perhaps reflecting on the passage of time, as your brain continues its gentle adaptation. Proteins are synthesized, synapses adjust, glial cells support metabolic health. Even in stillness, growth occurs at microscopic scales. There is no abrupt halt to this quiet renewal. Aging also involves structural changes in gray and white matter. Some cortical thinning is common, yet functional compensation often maintains performance. The brain's network organization becomes more distributed, drawing upon multiple regions to accomplish tasks. But flexibility remains a hallmark. Throughout life, meaning accumulates. Experiences leave patterns in neural circuits, shaping perspective. Emotional resilience may deepen. Connections between memory and identity strengthen. The brain carries stories within its pathways, stories of learning, love, loss, and curiosity. And as this lifelong process continues, it invites a final reflection, stepping back to see the brain not only as a structure that changes across time, but as a vast and luminous universe in its own right, filled with countless connections shimmering quietly in the dark. If you step back now, gently, from the details of cells and circuits, from neurotransmitters and networks, you may begin to see the brain in a different way. Not as a collection of parts, but as a vast inner cosmos. Within the curved shelter of your skull lives a structure containing billions of neurons, each forming thousands of connections. The total number of possible pathways exceeds anything easily imagined. And yet, it all unfolds in silence. Each neuron is small beyond ordinary perception, but together they create something immense. If you could map every connection, you would see a dense web branching and crossing in layered complexity. Scientists call this map the connectomy, an ever-changing record of how cells link together. It is not fixed. It shifts subtly with experience, sleep, and time. The universe within you is dynamic. Electrical impulses move along axons like brief flashes of light, though far smaller and quieter than lightning. Chemical messengers cross synapses, carrying signals from one cell to another. These processes occur in milliseconds, yet they form the foundation of perception, memory, and identity. Each thought you have is supported by coordinated activity across distributed regions. No single neuron holds a memory alone. It is patterns of activation across networks that give rise to awareness. The brain's energy demands reflect its constant activity. Though it represents only a small portion of your body's weight, it consumes a significant share of oxygen and glucose. This steady metabolic flow supports the maintenance of ion gradients, the release and recycling of neurotransmitters, and the repair of cellular structures. Even in sleep, this universe does not go dark. It shifts into different modes of operation, but the hum continues. Within this cosmos are regions shaped by evolution across millions of years. The brainstem, ancient and steady, regulates heartbeat and breath. The limbic system processes emotion and memory. The cortex, folded and expansive, supports language, reasoning, imagination. These layers are not stacked like separate floors. They are interwoven, communicating constantly. When you reflect on your own life, you are observing activity within this universe. Self-awareness arises from networks that integrate sensory input, memory, and emotional tone. There is no central observer seated at a control panel. Instead, consciousness emerges from the interplay of many regions working together. It is a process rather than a place. The brain also anticipates. Predictive models form in the cortex, generating expectations about incoming sensory information. When reality aligns with prediction, perception feels smooth. When it diverges, the signals adjust internal models. This predictive coding allows efficient interaction with the world. The universe within you is not passive. It is continuously forecasting. Plasticity ensures that this cosmos remains responsive. Synapses strengthen or weaken based on use. Myelin adjusts to support frequently traveled pathways. Even glial cells contribute, regulating chemical balance and clearing metabolic byproducts. The architecture is alive, maintained through constant microscopic labor. Across a lifetime, experiences accumulate as patterns of connectivity. Memories are not stored like files in a cabinet. They are distributed across networks, reactivated through coordinated firing. When you recall a childhood moment, fragments of sensory cortex, emotional centers, and associative regions activate together. The past briefly glows again within the present. Imagination draws upon the same networks. The brain can simulate possible futures by recombining elements of memory. This capacity supports planning, creativity, and empathy. The inner universe projects scenes not yet experienced, testing possibilities in safe rehearsal. These simulations influence decisions long before action unfolds. Even silence contains structure. When you feel at rest, resting state networks maintain gentle coordination. Patterns shift between focused attention and inward reflection. Brainwaves rise and fall in steady rhythms. Chemistry adjusts in response to light and darkness. All of it occurs beneath conscious control. The scale of this activity is astonishing. At any given second, trillions of synaptic transmissions occur across your brain. Each transmission is brief, yet collectively they sustain a continuous stream of awareness. If you imagine each thought as a star briefly glowing, then your mind becomes a sky of flickering constellations. Some patterns recur, forming familiar shapes. Others appear once and fade. There is no need to grasp each star. The universe does not require you to catalogue its lights. It shines and shifts whether or not you observe it closely. Understanding this can be calming. You are not responsible for orchestrating every signal. The system operates through feedback loops and self-regulation honed by evolution. Sleep will soon dim certain constellations and brighten others. Conscious awareness may recede, but neural activity continues in different configurations. Memory consolidates. Emotions integrate. Metabolic waste clears. The cosmos cycles through phases like night and dawn. You are both inhabitant and expression of this universe. The sense of self arises from its coordinated dance. Yet you are not required to control the dance. There is a quiet intelligence embedded in these networks, guiding adaptation and balance. As this journey gently comes to a close, notice how wide and still the world feels. So many thoughts unfolding quietly, whether we notice them or not. You don't need to keep their names or pathways. They've already done their work. If you enjoy these slow explorations, you can support the channel in any small way that feels natural, so others can find this calm too. But right now, there is nothing to decide and nowhere to go. Let your thoughts soften, your muscles relax, and the night carry you onward, one peaceful breath at a time.